Hello everyone, and welcome back for another biology video. And today we are starting in on chapter 3, and we are going to start talking about the important macromolecules that make up cells and biological systems. So if you kind of look at the topics rundown here, you can see we're going to talk about the four major types of macromolecules: carbohydrates, lipids, proteins, and nucleic acids. But in this introductory video, we are going to discuss how these types of molecules are built and then deconstructed and destroyed. So we're going to use this video as an opportunity to learn what these four different types of molecules all have in common with each other, and that's going to make our study of each individual one easier later on. So let's go ahead and get into this. So we discussed in our last video of chapter 2 that living matter tends to contain a lot of the schnapps elements that we've talked about before. Remember, carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. So we also distinguished organic, or inorganic molecules apart from the living, non-living issue, right? So what we decided is that those are not really the same things, right? So living cells, living tissue, is made up of both organic molecules and inorganic molecules, right? So organic molecules we defined as any molecules that contain both carbon and hydrogen. So living cells need to be made up of organic molecules like the ones we're going to talk about in this chapter, but also inorganic molecules like water. And we had a whole video in the last chapter detailing why water is so very, very important. Now, just to give us a little bit of a review, in case it's been a little while since you watched the last video, organic molecules that are found in cells tend to have a carbon-carbon skeleton, like what you see down here. And what we noted about this carbon skeleton is that it's got all these different vacancies that can potentially be filled in with a number of different atoms or functional groups. Now, the first thing we want to review here is the actual chemistry of carbon itself. So you'll hopefully remember that each carbon atom can form a total of 4 single covalent bonds, and that's really, as we mentioned in the last video, that's going to give us a lot of diversity in terms of what types of functional groups and atoms that we can kind of attach to the carbon skeleton in order to make a number of different types of macromolecules. So what we're essentially going to see is that when we compare carbohydrates to lipids to nucleic acids to proteins, they all essentially have this carbon-carbon backbone or skeleton or whatever you want to think of it as, but what different types of functional groups that get attached and therefore what types of chemical properties these biomolecules have, it's going to be a little bit different. So here you can see a little bit of a review in terms of the different types of functional groups we're going to be talking about. The ones we spent most of our time focusing on last time were the hydroxyl functional group, which is going to impart a lot of polarity to whatever skeleton it's a part of. We talked a little bit about carbonyl and carboxyl groups, so these are going to provide some polarity as well. The amino functional group we talked about, the phosphate group we talked about, the methyl functional group we talked about, which is going to impart some non-polarity and some hydrophobicity. We didn't really talk about the sulfhydryl functional group, but we will talk about that later when we specifically start talking about proteins. That's something you really only see in proteins. So like we said, if you have a empty carbon skeleton like what you see before you, we can fill in those vacancies with any of these different functional groups, and the functional groups, of course, have predictable chemical properties such as polarity or electrical charge or acidity or alkalinity, and these properties will be imparted to the macromolecule as a whole if the functional group gets attached to that particular skeleton. Now, if we want to kind of give you a little bit of an example of this, we talked about the issue of solubility in water. And we said that one example that we looked at back in chapter 2, the heptane molecule, where it's just a carbon skeleton that's just completely covered in hydrogen, we said that molecule is totally nonpolar and it's going to be very hydrophilic. So it's not going to dissolve in water very well. So what we've done here is we've taken that same molecule, except this time we plucked off 4 of those hydrogens and replaced them with a variety of different functional groups. So I want to illustrate what these functional groups are going to do to this molecule in terms of improving this molecule's solubility in water. The first one we can see here is an amine functional group. This is what we sometimes call a basic functional group, meaning that it's going to want to take on an extra hydrogen atom, which is what bases do, and in doing so, it's usually going to be positively charged at a neutral pH value. And by neutral pH, I mean a pH of somewhere around 7, if you remember that from Chapter 2. So if you have a macromolecule that has one of these amine functional groups, like what we're going to see in proteins, just be aware these amine functional groups are usually going to have a pretty solid positive charge on them. This hydroxyl functional group you can see right here is not going to have a full electrical charge, but it is going to be very polar. So it's going to be somewhat like water in the fact that it's going to have partial positive and negative charges associated with it. But it's still something that water's going to want to grab onto, just as is the case with this amine group here. The carboxyl functional group is kind of the opposite of the amine functional group. This is an acidic group, meaning it's going to want to give up a hydrogen atom. And if it does that, it should have a pretty strong negative charge at a neutral pH value. So if the amine functional group is positively charged, the carboxyl is going to be negatively charged, whereas the hydroxyl is just polar. And then the phosphate group you can see down here, this one is going to be negatively charged pretty much all the time. So it's almost always negatively charged. So again, it's something that water can grab onto. So in all 4 cases here, we have given water something to interact with. So this molecule, as you're looking at it right here, is much, much, much more hydrophilic and soluble in water than the heptane molecule that had nothing but carbon-hydrogen bonds. So the whole bottom line here, if you want to take something away from this, is that if you can provide the backbone with an increased number of polar or hydrophilic or electrically charged functional groups, you're going to make that molecule more and more and more soluble in water. And if you do that, you're potentially increasing how significant a role it's going to play in biology, because as we detailed in the last chapter, biologically important molecules tend to be hydrophilic. Not all of them, but they tend to be hydrophilic. Okay, so let's introduce a new concept here. How are we going to build a macromolecule? And more particularly, how are we going to build that carbon-carbon skeleton, or backbone, if you will? So if we're going to build a molecule, the way we're going to do it is we're basically going to treat it like building with Legos or building with building blocks. We're going to take a bunch of little smaller building blocks, which are called monomers, and we're going to attach them together to form something much bigger and much longer, which is called a polymer. So this polymerization we're dealing with here is how we build all of our macromolecules, our proteins, our carbohydrates, our lipids, and our nucleic acids. So we take a number of smaller molecules and we start linking them together by forming covalent bonds between them, so now they are solidly attached to one another, and we just keep doing this over and over and over again. And because those monomers already have particular functional groups attached to them, we end up with a long carbon-carbon-carbon skeleton that has functional groups kind of sticking off to the side. So the way that we build our polymers is by performing a type of chemical reaction called dehydration synthesis. So what we have here is we have our monomers, and we are going to add another monomer to the growing chain by this type of chemical reaction, which again we call dehydration synthesis. The reason why this is called dehydration synthesis is you can actually see that an element of water is lost between the 2 monomers that we are about to attach together. So this monomer right here has a hydrogen atom sticking off of it, and this monomer here has an OH sticking off of it, so H2O. So when we form this new covalent bond between these 2 monomers, we lose that element of water, the 2 hydrogens and the oxygen, and a water molecule is generated, which is why we call this dehydration synthesis. We pull out a molecule of water in the process of connecting these 2 monomers with a covalent bond. So that is how we build polymers. We just keep adding new monomers to the growing polymer chain, and there shouldn't be too much of a limit on how long our polymer can be. But every time we perform one of these reactions, we lose a molecule of water, and we gain a covalent bond that links two monomers together. So that is how we build a polymer. Well, it turns out we can also pick a polymer apart. We can basically destroy it by breaking the covalent bonds that join the monomers together. And that type of chemical reaction that we're having there is called hydrolysis. So we have here a polymer that consists of 3 monomers stuck together. And if we want to break this covalent bond so that we obtain a single monomer all by itself, we have to use a molecule of water to break that covalent bond. So that's why this is called a hydrolysis reaction. A molecule of water is used to break the covalent bond that joins 2 monomers together. So these two reactions are almost exactly the same. It's just one is the forward reaction and one is the reverse reaction. You might remember that terminology from chapter 2, the idea of a forward reaction and a reverse reaction. We either build the polymer or we break it apart. So you can see the same sort of thing happening here. We're joining 2 monomers together by dehydration synthesis. And then if you want to take the opposite reaction, the reverse reaction, you take the covalent bond that joins the 2 monomers together and you break it apart and use a water molecule to get that done. So the important thing here is that whether your macromolecule that you're talking about, whether it's a protein, a lipid, a nucleic acid, or a carbohydrate, the manner in which we build our polymers or break our polymers apart, it's exactly the same. We use these same 2 chemical reactions for building proteins and lipids and carbs and nucleic acids, and we use the same reactions for breaking them down. Dehydration synthesis for building, hydrolysis for breaking apart. So these reactions are exactly the same no matter which of these 4 types of macromolecules you happen to be talking about. The only difference is in the identity of the monomer. What makes a protein different from a carbohydrate is not how they are built, it's what kind of building block do you start with, as we are going to see. So another important thing that we're not really going to appreciate, I think, until probably chapter 6 or so, when we start talking about thermodynamics and cellular energetics, is that these reaction rates, meaning how many reactions you get per minute or per hour, the unaided reaction rates for dehydration synthesis and hydrolysis, they're very slow. These are very, very, very slow chemical reactions if you're not giving them some help. So what we'll see later on when we can appreciate this a little bit better is that the cell has these catalysts. A catalyst is a substance that speeds up a chemical reaction. The cell has protein-based catalysts called enzymes that are specifically there to speed these reactions up when necessary. Okay? So if we kind of look at what our different types of macromolecules are, we want to focus on what makes them all similar, which is that they are all polymers that are built using dehydration synthesis, but then also what sets them apart. And what sets them apart is what kind of monomer is used to make the polymer. So if we start by looking at carbohydrates, carbohydrate, like the starch that you see here, this starch is just a big polymer of little individual sugar molecules called monosaccharides. The monosaccharide is the monomer that is used to build the carbohydrate polymer. That is different from a nucleic acid. A nucleic acid is a polymer that is built by attaching individual nucleotides together. A nucleotide is the monomer for building nucleic acid polymers, like this DNA that you see here. So if we compare and contrast carbohydrates and nucleic acids, they're similar in the way that they are built by sticking monomers together to build a polymer, but they are very different from each other in the sense that the monosaccharide monomer is very different from the nucleotide monomer. So carbohydrates are polymers built by attaching monosaccharide monomers together, and nucleic acids are polymers built from attaching nucleotide monomers together. And then if we consider proteins and lipids, we can see pretty much exactly the same thing. A protein is a polymer built by attaching individual amino acid monomers together. So this time the monomer for a protein is an amino acid. It's not a nucleotide and it's not a monosaccharide. It's a completely different type of monomer that is specific for proteins. And then for lipids, we build our polymers, and this is a little bit more complicated, and I'll kind of clear up any confusion here because these are not true polymers. But if there were going to be a polymerization here, we would be building our polymerization with a monomer called a fatty acid. But again, I will clear that up in a later video where we specifically talk about lipids. So if you kind of consider what our different types of macro or biomolecules are made of, carbohydrates are made up of sticking monosaccharides together to build a polysaccharide. Lipids are built by sticking together glycerol with the fatty acid monomers to make a fat. Proteins are built by sticking amino acids together to make a polypeptide. And then nucleic acids are built by sticking nucleotides together to make DNA or RNA. So consider the fact that for each of these different types of macromolecules, we are building them all exactly the same way by forming a polymer out of sticking monomers together, but these different types of molecules end up being fundamentally different because of the difference in terms of what monomer we start with, what your building block initially is. Okay, so that's going to do it for this introductory video for Chapter 3. Thanks, as always, for your attention. You can see for the next 4 videos, we are going to give each of the 4 types of macromolecules their own time to shine. So our first video is going to be on carbohydrates, and then we will have a separate video on lipids, a separate video on proteins, and then finally a separate video on DNA and RNA. Thanks again, and I will see you next time. So long.